Yes, hi everyone, and just some words to introduce our speaker, Alison Bell. She is a professor in the Department of Evolution, Ecology and Behavior, and is the leader of the Gen Networks in Neural and Developmental Plasticity team at the Carl Woese Institute for Genomic Biology at University of Illinois. And she earned a PhD in population biology at the University of California, and then was a National Science Foundation postdoctoral fellow at the University of Glasgow in Scotland. And her research is focused on individual differences in behavior using mainly 3-spine stickleback fish as a model organism. And she integrates approaches from neuroscience, genomics, and evolutionary biology to understand why individuals differ from both proximate and ultimate perspectives. And today she will talk about molecular mechanisms of behavioral and transgenerational plasticity in sticklebacks. A very exciting program, and I think you can start, Alison. Thank you very much.
Excellent. Thank you so much. Well, thank you, Emilienne, for the opportunity to participate in this seminar series. This is one of the few good things to come out of the pandemic, is all of these conversations that we're now able to have over long distances. I realized it's late there in France. It's early in the morning here in Illinois, but here we are together. So thank you very much for the opportunity to be here. Okay, so today I'm going to be telling you about behavioral plasticity and molecular mechanisms of plasticity within and across generations. And so my interest is primarily in behavioral traits. And behavior, I think, is interesting for many, many reasons. But one of the reasons is that behavioral traits are sort of the canonical complex trait. They're typically influenced by many, many interacting genes of small effect. They're notoriously sensitive to the environment, and in the case of social behavior, sensitive to the social environment. So indeed, that's often what's most interesting about behavior, is that it's very responsive to the environment. And there's tremendous variation in what behavior actually looks like across organisms, and also what behavior looks like in different contexts or different situations. So birds singing, flies fighting, honeybees communicating, etc. And this, of course, has long fascinated biologists. So over the past 20 years or so, the scientific study of animal behavior has discovered genomics and transcriptomics in particular. So for example, there are a growing number of studies interested in asking about how the genome responds in terms of gene expression to social interactions in the environment. So what happens in the brain of, say, a zebra finch when it hears a song, or in the brain of a female swordtail fish when she's presented with a potential mate, or in the brain of a fruit fly during an aggressive interaction. And then people have been thinking hard about what we can learn from these sorts of transcriptomic responses in brain. And I think there's really two kind of key insights that have emerged from these studies over the past 20 years or so. And the first is that we now know that genes influence behavior via interacting with one another in gene regulatory networks. There is rarely, usually not, a simple one-to-one mapping of gene to behavior, but instead genes operate through networks of interacting genes in the brain. The second is that the genome is incredibly environmentally responsive via dynamic changes in gene expression in brain. This used to be surprising to us, let's say 20 years ago, since we thought that the main dynamic response system was the neural response system in brain. But we're now appreciating that the genome is also highly environmentally sensitive. So both of these insights, I think, have some pretty profound implications for how we think about the relationship between genes and behavior, and they sort of buck the way that genes and behavior is often and still tends to be taught. Namely, we now know that there's not a usually clear causal connection between single genes and single behaviors, but the pathways are a lot more circuitous, not deterministic, and environmentally sensitive. And this way of thinking also has profound implications for getting us then out of some simplistic kind of dualistic nature-nurture dichotomous thinking that has really plagued our understanding of behavior in the context of biology and evolution. But given how expensive many of these studies are when we're talking about using something like RNA-seq, for example, to measure gene expression, typically studies just measure gene expression at one point in time. And if you're really lucky, they might be measuring expression in more than one brain area, but usually it's often actually in whole brain. And with only one time point and one brain area, you're really limited in what you can conclude because gene expression is really dynamic. And by just taking a snapshot, you're really just getting one little small glimpse into the underlying dynamics. So I'm making this point to drop a hint at a theme that's going to be coming up again and again in this talk today, namely about the insights we can get by thinking more seriously about timescale and dynamism. So for today, I'm going to be telling you about two case studies on behavioral plasticity, molecular mechanisms of plasticity. The first having to do with territorial aggression, and then on to parental care, and then I'll go on to talk about within and transgenerational plasticity. So the star of today's show, as Amelia mentioned, my lab focuses our work on 3-spined stickleback fish, who may be familiar to many of you. If you've taken an evolution class recently, sticklebacks have become prime examples of rapid evolution and a great model system for underlying the genomic basis of adaptation. But sticklebacks are also really wonderful for studying behavior, of course, because there's a long tradition of studying stickleback behavior, both in the field and in the lab, dating back to the classic studies by the early ethologists. So here's some really basic biology, reproductive biology about this species that's going to be important for the rest of the talk. So sticklebacks are seasonal breeders. So during the breeding season, which is actually just starting right now, males will start to defend nesting territories, build a nest, and attract females to come and mate with them in their nest. Fertilization is external, so female swims through the nest, releases her eggs, he swims after her, fertilizes the egg, and then she buzzes off, and he does all of the parental care. So parental care is really important for male fitness. So the developing embryos will die if they don't get care from their fathers. So he spends a lot of time oxygenating the eggs by fanning them. And then one of the really, really important things that he can do is to defend his nest because caviar is delicious. And so this is just a video to show, this is a male stickleback. He's the guy with the red chin and the blue eye who's defending his nest, which is this pile of algae right here from this band of California roach that are attacking his nest. So territorial defense and parental care are some of the most important things for male stickleback fitness. So if a male fails to successfully defend a territory, he will not obtain a mate and he will not produce offspring. So effective territory defense is absolutely necessary if a male has any chance of having reproductive success. Okay, so if we step back and think for a moment then about what is happening when a male stickleback, or really any organism for that matter, is confronted by a stimulus like, say, a conspecific, you can imagine there's probably a lot that can't just be captured by a single snapshot in time, let's say after a behavioral interaction, or let's say by comparing, say, extreme, aggressive versus non-aggressive individual types. So you might imagine that when an animal is presented with a conspecific, there are elements of perceiving the conspecific, then responding to that conspecific behaviorally, maintaining a behavioral response, and then once that interaction is over, recovering from it and preparing for the future. So it seems plausible, I think, that there are probably waves of transcription, let's say, that are associated with all of these different components or elements to behavioral plasticity, but we probably can't learn that much about that process from just a single cross-section in time. Moreover, by just measuring RNA, so the transcripts of our gene expression, it's hard to make inferences about gene regulation or how all of this is working at a molecular level. So it's useful to have other types of information, other types of data. So given the dynamic nature of behavior and transcriptomics, it's likely that we can learn a lot about behavioral plasticity via something like a time series sampling approach. So I'm going to tell you about an experiment that was done by Abbas Bukhari, who was a PhD student in the lab and now a postdoc as part of a collaborative project funded by the Simons Foundation. So we investigated the neurogenomic response to a social challenge. So this was a territorial challenge in the lab where nesting males are housed individually in separate tanks, and then we can confront a nesting male with an intruder who's been confined to a flask, measure that male's behavioral response to this simulated intrusion, and then sacrifice males at different time points after this simulated intrusion event. So specifically in this experiment, we sacrificed males at 30, 60, or 120 minutes after the territorial intrusion, and then used RNA-seq to measure gene expression in 2 regions of the brain. As a control group, we had a set of males that were presented just with an empty flask. So this was just a control for, let's say, the effects of splashing or a novel object or something like that into the tank. So in addition to measuring gene expression in these 2 different brain areas, we also used ChIP-seq to measure changes in chromatin accessibility at different time points and just in diencephalon only. And we used a ChIP-seq using H3K27AC as a marker of chromatin accessibility. Okay, so just to show you some of the initial data that came out of this project, here are some Venn diagrams showing the number of genes that were differentially expressed between the control and the experimental group. So that's males that were confronted by a male in a flask versus just an empty flask at these different time points following a territorial intrusion. So 30, 60, and 120 minutes, and so shown separately for the 2 different brain areas. So the main pattern I want to just draw your attention to is check out the relatively large number of genes, so that's what each of these different numbers represent, that is unique to each time point, that is not shared across time points. So there's actually relatively few genes within a brain region that are shared across time points within any given brain region. So this is an immediate clue right here that the neurogenomic response to a territorial challenge is very dynamic. And it's also very sobering to those of us who do these sorts of experiments that you get very different results depending on the timing of sampling. So most studies, as I mentioned, we typically just say sacrifice and measure gene expression, let's say 30 minutes, because that seems like a reasonable time. But if you had measured at 60 minutes or 120 minutes, you would get very, very different answers. Okay, so to try to get a sense or actually try to understand more about this dynamism in response to a territorial challenge, Abbas built a generalized linear model that tested for the interaction between time, that's 30, 60, 120 minutes, and treatment, control versus experimental, in order to identify genes whose expression profile changed over time in response to a territorial challenge. And he did this separately for the 2 different brain areas. He then used hierarchical clustering on those genes, which managed to group them actually into different clusters with particular expression profiles over time. So that's what's shown here in these different boxes. There were some clusters of genes, for example, that were downregulated at 30, upregulated at 60, and then downregulated even further at 120 minutes following the challenge. There are other clusters of genes that are up and then down and then way up. So this is just showing the results for diencephalon, but we did the same thing for telencephalon and found similar patterns. So each of these lines here represents a different gene, and this hierarchical clustering method was able to identify clusters of genes with similar expression profiles over time. Okay, that's kind of cool. But Abbas then performed gene ontology enrichment analyses on these clusters and found that each cluster was characterized by a mostly unique, non-overlapping set of Go terms, which are shown here on the right. So I don't expect you to read this text, but what I want to draw your attention to is the pattern. So the columns here are the clusters, and the rows are Go terms. Notice that each cluster has its own set of Go terms that's associated with it, and those Go terms are not shared with any of the other clusters. So, for example, this cluster has one Go term associated with it that's not shared with any of the other clusters. And if you could read what this says, you could learn that these are all genes having to do with hormone activity. And in fact, genes in this cluster include things like CGA and prolactin. So I think what this analysis is telling us is about what is the molecular function of these waves of transcription that are following a brief social interaction. And those waves of transcription have particular functions. And I think this makes sense, actually, when you think about the biology of social behavior. There's sort of an immediate reaction, a transcriptomic response associated with the maintenance, recovery, et cetera. So there are some biological functions that peak early and then subside, while others peak at 60 minutes, while others peak hours, let's say 120 minutes later following a social interaction. So Abbas then sent all of these data to our collaborator, Sriram Chandrasekaran, who had developed a transcriptional regulatory network algorithm that uses the expression of transcription factors and their targets to build a network describing their associations. So Sriram mapped those clusters that Abbas had identified onto the network, and the results are actually pretty neat. There's a ton of information on this slide I don't expect you to grasp here, but what I want to draw your attention to is just a few interesting patterns. So the way to read this is that each of these different circles or nodes in the network represents a transcription factor, and edges connect transcription factors whose expression are statistically correlated with each other. All of these different pieces of pie within a node represent a different cluster. So clusters of genes in diencephalon are in orange, and in telencephalon, they're purple. There's a lot to take in here. For the purposes of this talk, the main point that I'd like to make is that we were able to integrate this gene regulatory network with that clustering analysis I just showed you. And we identified transcription factors that regulate each of the different clusters of genes. And if you could stare carefully in here, you might see that there's this group of transcription factors that all have a little piece of purple pie in the same spot. So that's suggesting that these are all transcription factors that are regulating the same cluster of gene, and their expression is correlated with one another. So this was the nice little benchmark test that we're getting kind of consistent results. I mentioned that we also measured changes in chromatin accessibility following a territorial interaction. So here's just some of the summary results in the Venn diagram. Each circle represents the number of genes in a particular set. So this blue circle here represents the number of genes that were close to peaks that were differentially accessible in the control versus experimental group. So this is when the magnitude of the peaks differ between those treatments following a territorial challenge. And this greenish circle is the number of genes that were differentially accessible at 30 minutes, and the red circle are the number of genes that were significant by that time for treatment interaction term that we used in the clustering analysis. So we found that many of these genes that were significant for this time by treatment interaction term were near areas of chromatin whose accessibility changed in response to an intrusion, especially 2 hours afterwards. So just thinking here about the raw numbers of differentially accessible peaks, I must say that we were actually a little bit surprised by this result because we weren't expecting to see dramatic changes in chromatin accessibility following a 5-minute social interaction. But I think it actually is pretty cool that it looks like a fairly brief social event was sufficient to cause pretty dramatic changes in chromatin accessibility. And that's something that we often think of that changes pretty slowly. So Abbas integrated these changes in chromatin accessibility with the gene regulatory network analysis, with the clustering analysis, and found some really neat patterns. I'm just going to give you one example here. So here's an example, PPARG, which is a nuclear receptor. These are tracks for the chip results. So the different rows are for the 30-minute control, 30-minute experimental, 120-minute control, 120-minute experimental samples. And then this is the track for H3K4ME3 to mark the promoter of this gene. And what I want to draw your attention to is there's this peak in the 120-minute experimental track that's right over the promoter, indicating that the promoter of PPARG became more accessible at 120 minutes. Going back to the RNA-seq data, separate experiment, different animals, different year, here's the expression pattern of PPARG over time. It was upregulated in experimentals at 120 minutes. We then looked for PPARG in the gene regulatory network, and here is the expression pattern of PPARG's targets showing fold changes. And the expression pattern of the targets of PPARG maps the expression pattern of PPARG itself. So it's not differentially expressed at 30 minutes, down at 60, and up at 120 minutes. So what was kind of neat about this is that we were able to connect then changes in chromatin accessibility to transcription factors that had been identified in this gene regulatory network analysis, to gene expression and the targets of those transcription factors. And while this type of analysis had been done a lot, let's say, in biomedical literature or in studies on development and things like that, this is showing that these same properties of gene regulation, namely about changes in chromatin accessibility, transcription factors, and their targets, can also apply to something like socially induced changes in gene expression. So what do we conclude from all of this? So first, I think that this time series sampling has taught us something different about behavioral plasticity that you simply can't get from a single snapshot or cross-section in time. It allowed us to capture waves of transcription in response to a social interaction. And we think this is telling us about the molecular basis of behavioral plasticity, that there are different waves of transcription that have different functions. And that makes sense when we think about the biology of plasticity. Second, these data show that a brief but highly consequential social interaction is sufficient to cause transcriptomic changes that can persist for hours afterwards. So while most studies I mentioned typically measure transcriptomic responses following social behavior for, let's say, 30 minutes or so after the social behavior, we still saw lots of changes in gene expression for 2 hours after this 5-minute territorial intrusion. Moreover, parts of the genome were becoming more accessible at 120 minutes, which suggests that genes in these newly accessible areas are poised to become activated. So we might have seen changes in gene expression even longer. This is something, when you do an experiment, you're like, I wish that I had waited, another 24 hours or something, because who knows for how long we would have detected differences. And from my point of view, as like a behavioral ecologist who likes to spend time actually watching what animals are doing in nature, when you actually watch sticklebacks in the field, they are constantly interacting with their neighbors to negotiate territory boundaries. And it kind of blows my mind to think about what happens during each of those social interactions, let's say, while they're negotiating territory boundaries. So this is probably happening over and over and over again. So finally, our results suggest rapid and dramatic epigenomic plasticity due to social interactions. So the chromatin data, I think, were really quite surprising to us because it changed faster than conventionally expected, within hours, and it also changed pretty dramatically. So we have some cases here I didn't show you, but actually regions of the genome that were inaccessible but became accessible following an intrusion. And part of the reason why this is sort of surprising to us was that because there weren't that many examples at the time of this study in the literature showing changes in chromatin accessibility. There had been a few studies looking at things like drug effects or changes due to cancer or something like that, or during embryonic development, exposure to cocaine. But this was a 5-minute social interaction. And so we're seeing these really dramatic changes, epigenomic changes following pretty brief and brief social behavior. So what is going on here? We don't know. I can tell you that right now, but what do we make of these results? So why do all of these changes persist for a long time, and what do they really reflect? So this is the reigning hypothesis, and it is a video showing an interaction between two sticklebacks in a tank where they're negotiating over their territory boundary that's indicated by this red line here. So in nature, sticklebacks are constantly engaged in interactions with their neighbors and intruders over the boundaries of their territory. And those boundaries are often being updated. And if you think about it, there's probably a lot of learning that's going on during an intrusion. Territory holders are getting information about the spatial boundaries of their territory, the competitive ability of their neighbors, and their own resource holding potential, for example. So indeed, territorial animals often improve their ability to detect and repel intruders with experience. And the behavioral literature is rife with examples of experience-dependent changes following a territorial challenge, such as the prior residency advantage, winner effect, dear enemy, etc. So we think spatial learning is particularly likely here because fishes, we know, sticklebacks actually will actively patrol sites where they've had a previous encounter with an intruder. So this is relevant in this case because we actually detected a strong signal of learning-related genes in this data set, especially at 120 minutes, and epigenetic changes are being increasingly recognized and is important during memory formation. So our current hypothesis is that this brief territorial challenge triggered the expression of learning-related genes and that changes in chromatin are associated with the formation of memories about where that social interaction occurred. Okay, so that was part one about territorial aggression. Now I'd like to tell you about a second case study about behavioral plasticity and molecular mechanisms having to do with parental care. So like territorial intrusion, parental care is also a very dynamic kind of phenomena, but actually tends to occur over longer timescales than something like behavioral plasticity in response to an intrusion. So we were interested in applying the same kind of approach, thinking about dynamic changes in gene expression in the brain to another really important social behavior, namely parental care. So parental care is really dynamic. So if you think about it, all say caregiving animals go through a predictable kind of series of stages. They might start in, let's say, a non-breeding state, and then enter, say, a pre-care preparatory state like territory establishment and courtship. And once they've got their home all set up and mating has happened, the parents may then start to provide care for their embryonic offspring. This could be something like incubation in oviparous animals or pregnancy in mammals. Then after the offspring are born or hatch, parents might also enter a postnatal care stage where they provide postnatal care in the form of things like provisioning or offspring defense. So clearly there are really different behaviors going on at all of these different stages of care. So we might expect each stage of care to have its own kind of unique genomic signature. Also, though, in order to proceed to the next stage, the parent has to have completed the preceding one. So there's an important temporal ordering to these dynamics. So we might imagine that there could be facilitating effects of certain hormones or neuropeptides that the animal has to experience in order to move through the stages. I think it's also interesting to think about how some behaviors kind of get added to the behavioral repertoire as parents go through these stages. So like starting to feed offspring, for example. But there are also some elements of continuity across these stages, like the presence of a nest, for example. So we wanted to see if we could see a signal of any of these sorts of changes. So continuity across stages, unique elements to each stage, carryovers at the neurogenomic level. So what exactly does parental care look like in a sticklebacks? Here I get to show you a video of a male stickleback. This is viewed from above. This green thing here is his nest. This is in the lab. And so this is a male who is actually providing care for a newly hatched fry. And if you look closely in the video, you can see this that thing right there, that is a little free swimming fry that he's picked up in his mouth and spat back into the nest. So I mentioned that male sticklebacks, they provide care for the developing embryos by doing things like fanning them and defending them. But then after the embryos hatch, he actually continues to care for his newly hatched fry, doing things like retrieval behavior as the fry start to become free swimming and start exploring on their own. He'll chase after them and spit them back into the nest. Eventually, he can't keep track of them anymore and they disperse. Okay. So we did an experiment where we sampled then male sticklebacks with 5 males per stage at 5 different stages of care. So this again was work that was done by Abbas Bokhari, pictured here. So we sampled males for gene expression profiling with RNA-seq in 2 different brain areas again, telencephalon and diencephalon, during 5 different stages of care, and then compared them relative to a non-breeding control group of males with 5 males per condition. So specifically, we sampled males that had a nest, males that had eggs in their nest, and males at 3 different points in time while the eggs were just starting to hatch. And so the rationale for this is that hatching takes place sort of over the course of a day. The eggs just start to hatch in the morning, about half of them hatch in the middle of the day, and the clutch is fully hatched late in the day. And we sampled this way because we were interested to see males' initial reaction to all of a sudden being responsible for these little free swimming fry. So males were compared relative to a control group of reproductively mature males that did not have eggs and which were sampled at the same time of day to control for circadian effects. So the way that Abbas did this was to identify differentially expressed genes between each of these different stages and the control group. And then he looked for similarities and differences between the differentially expressed genes across stages. And so specifically, we're interested in knowing about 3 different types of genes. So first, because males do really different things behaviorally at each of these different stages of care, we thought that there might be some genes that are unique to each stage of care, much as in the same way that some behaviors are unique to each stage. Second, behaviorally, there's also some similarity across stages, like the presence of a nest, for example. So we wanted to know if there are genes that carry over across stages that might resemble something like what's happening at the behavioral level. And then third, we thought that there might be genes that get added as a new behavior or a new stimulus is sort of incorporated, like all of a sudden the presence of eggs in the nest, for example, or having to care for a newly hatched fry. And given that males can't go on to one stage without having gone through the preceding one, we thought that some genes might pop up in one stage and then go on to carry over into the next stage. And so we wanted to see whether any of these changes in behavior and the availability of, say, exogenous stimuli across stages could be reflected at the neurogenomic level. So do we see evidence for our first question about whether there are genes that are unique to each stage? So the answer is yes. As I mentioned, males do really different things behaviorally at each of these behaviorally defined stages of care. And so we wanted to see if that was reflected at gene expression in the brain. So for simplicity, I'm just going to focus on one brain region here, diencephalon. Same patterns hold for telencephalon. So to get at this question statistically, Abbas tested whether there were genes that were differentially expressed between the control and experimental group in a given stage, but not at any other stages. And he used a pretty strict statistical threshold for calling a gene unique to a stage to guard against the possibility of false negatives that say like a gene was differentially expressed in a different stage, but just barely missed the cutoff or something. So these are showing heat maps with genes as rows and stages of care, nest, eggs, early, middle, late hatching as columns. And here you can see that there are genes that are differentially expressed during the nest stage, but those same genes are not differentially expressed in other at other stages and so on. So yes, it does appear that there are genes that are unique to each stage. So they're popping up at one stage, but not another one. But what about similarities across stages? So behaviorally, there are some things that males do during all these stages, like maintain a nest. Do we see that reflected at the neurogenomic level? So to get at this question, Abbas looked for overlap between the differentially expressed genes across stages. And here we're plotting the magnitude of the p-value for the overlap between pairwise stages, such that basically bigger circles mean more sharing. So for example, here is the overlap or similarity between differentially expressed genes during the nest and the egg stage, nests and early, nest and middle, etc. And the pattern that I want to draw your attention to is that the size of these circles sort of gets smaller as you move further out in time. So in other words, stages that are closer together in time share more genes than stages that are further apart in time, which made us think that there might be genes that are carrying over across stages that are close in time, because stages close together in time were more similar. So the next question we were interested in has to do with whether or not starting a new stage initiates biological processes that can then carry over into subsequent stages. So once he gets eggs, for example, do those egg-related genes remain regulated the same way into subsequent stages? So to get at this question, Abbas looked for genes that were differentially expressed at a particular stage, but were not differentially expressed during the preceding stage. And then he asked what was the expression profile of those genes into the next stage. So a good example is here at the bottom. So these are genes that were differentially expressed during the middle stage, but were not differentially expressed in a preceding stage. And as you can see, they remained expressed in the same direction into the next stage. So the point I'd like to make with all these results is that considering the time course of changes in behavior prompted some questions about how and whether this is reflected at the molecular level in the brain. And one of the things I really appreciate about this analysis is that Abbas did was that it embraced the sort of dynamism of a behavior like parental care and illustrates how behaviorally motivated hypotheses can help us make sense of clearly a really complex process. I also think it's worth noting that there are probably a lot of other really important events in an organism's lifetime that can also be broken down into a series of stages, like the stages of dispersal or the stages of pair bonding or territory establishment, or for example. And breaking down the dynamics in this way captures some really interesting patterns that we could not have detected if we had just taken a snapshot. I think it also gives us some insight into the types of neurogenomic processes involved in regulating behavioral plasticity. So we're also starting to use some single cell transcriptomic data to get more insight into the cell types that are important for care. And we have some preliminary data using 10x technologies. So this is showing clusters of cells in the stickleback brain. And we actually found that there were 2 clusters of cells in this single cell data that were enriched, or that were basically expressing genes that were differentially expressed in our parental care data set. And what's even cooler is that it looks like genes that are associated with different stages of care can be isolated to specific cell populations. So therefore, not only were we able to detect a sort of neurogenomic signature associated with different stages of care, but we might be able to isolate those signatures to different cell types. So this is ongoing work. We're pretty excited about using single cell data in the future. So another really cool result, I think, from this study is that we found that many of the canonical genes typically associated with mothering were also differentially expressed in stickleback dad brains. So here's showing the profiles of the teleost homolog of oxytocin, isotocin across stages in both brain regions, in the experimental group, which is in red, and the control group in blue. Also estrogen receptor, prolactin. I love prolactin. It's especially interesting because it was named for its role in promoting lactation in mammals, but was actually probably serving functions related to caregiving in male fishes long before mammals were providing care. So these dynamics of hormone-related genes over time really reminded us of the hormonal dynamics of reproduction in mammalian females that are, of course, very, very well studied. And I think it's pretty cool that genes that are related to hormones that we typically associate with things like lactation and pregnancy, like progesterone and estrogen and oxytocin, are being differentially expressed in stickleback males. In other words, these genes are serving functions related to caregiving in a male fish. So these canonical genes really made us think more about whether we could formally compare the neurogenomic dynamics of parental care in a fish to maternal care in mammals. And right around the time when Abbas was doing this analysis, there was a paper published by Ray et al. That measured gene expression in 4 different brain areas in mouse and at a bunch of different stages, so during pregnancy and postpartum periods. So Abbas wanted to leverage this data set to ask to see if we could detect commonalities between genes that carried over across stages of care in stickleback, and whether they were also ones that were important for parental care in mouse. And he wanted to do this in an unbiased way, rather than just cherry picking the genes that our eye was immediately drawn to in our gene list. So this is obviously hard to do on a strict gene by gene level, because these are very different species and the genes are not necessarily homologous. So he did this test at the orthogroup level using software called OrthoDB. And so in stickleback diencephalon and mouse hypothalamus, there were 14 orthogroups which were shared between the 2 species gene lists in this parental care data set. And this overlap was greater than you would expect due to chance. Abbas here is plotting the p-values of the orthologous genes as a heat map. And so there are some interesting ones here, including things like BDNF, important for behavior and psychiatric disease. So this analysis is suggesting that there are some elements of the persistent, say, neurogenomic state of a paternal stickleback that is shared with the persistent neurogenomic state of a maternal mouse, which I think raises some interesting questions about similarities between maternal care and paternal care, and also about the evolutionary conservation of genes related to caregiving. It also prompted Abbas to pose the interesting idea that mammalian moms might actually be tapping into their inner fish during caregiving play on Neil Shubin's great book, Your Inner Fish. Okay, so to summarize this part of the talk, I showed you that the brain transcriptome is highly dynamic during parental care. We found some genes that carried over across stages, others that are unique to a stage, and some genes that got added to a male's neurogenomic state as he proceeded through stages of care. And we showed evidence that elements of the neurogenomic shifts during care in a fish are deeply conserved and are not sex specific. So to give you a sense of where some of this work on parental care in the lab is going, one of the things we're really interested in right now, this is just a teaser about this really cool ecotype of stickleback that provides a really exciting opportunity, I think, to study the evolution of parental care and its underlying mechanism. And that's this crazy ecotype of stickleback that occur in Nova Scotia. So these are the so-called white sticklebacks, where the fathers don't provide care. So as you can see in this picture, this is taken out in the field. This is a mound of algae, and these white things right here are white stickleback. So males of this ecotype turn white during the breeding season. But what we're actually most interested in about these male white sticklebacks is that they actually do not provide parental care. So after fertilization, instead of fanning the eggs and caring for them, males of the white ecotype pick up the eggs. That's what you're seeing right here. So this is his nest. He's picking up the eggs from his nest and he's spitting them out. And he's not providing any care at all. Now, those embryos seem to do just fine without care from their fathers, and it means that the males of the white ecotype can immediately go back into the breeding pool and start courting females again. So we have a bunch of ongoing projects on this really interesting ecotype that I'd be happy to tell you about another time. Okay, but for now, I want to tell you about a comparison of within and transgenerational plasticity. So there are a growing number of studies that many of you are probably familiar with if you interact at all with M and diverse species showing that when parents, typically mothers, are exposed to predation risk, that they can produce offspring with predator-adapted phenotypes. So, for example, exposure of gravid female crickets to predatory spiders caused females to produce offspring with heightened anti-predator behavior and higher survival in the presence of a predator. So similar types of patterns have been documented in a wide range of taxa, including in fish, lizards, snowshoe hares, aphids, Daphnia snails, and plants as well. And in only some of these cases do we actually know whether the effects of maternal exposure are adaptive, whether the changes in offspring actually improve survival. And most of these studies have really focused on the effects of predation risk to mothers, but we're now starting to appreciate more and more that fathers can also influence the development of their offspring through things like sperm-mediated effects or via their effects on mom or on their paternal behavior. So this kind of transgenerational plasticity could be adaptive because if parents can detect cues about the likelihood of predation risk for their offspring, and if they can prepare their offspring for living in a high predation environment, this could increase their offspring's chances of survival. And this kind of process could occur faster than genetic adaptation, avoiding some of the costs of both having to acquire information yourself in the form of developmental plasticity or within generational plasticity and producing the wrong evolved phenotype. And this kind of transgenerational plasticity might be especially helpful for adapting to new environments. So just to make sure we're all on the same page here, I'm defining transgenerational plasticity as occurring when the environment experienced by a parent influences the phenotype of their offspring. And the way that we typically study this is by comparing offspring whose parents experience different environments indicated by the different shading here, and then rearing offspring from the different parental treatments in the same environment. So this is all well and good. This is super interesting, but the biological reality is that an individual might be getting some information not only from its parents, but also via its own experience in the form of within generation developmental plasticity. And so an interesting problem, stepping back, is that animals in natural populations must decide how and whether to attend to information about risk from different sources. And those sources might not always agree with one another. For example, an animal might obtain information from their father that the environment is safe, while personal experience suggests otherwise. Maybe individuals should pay more attention to their own experience because it's more up to date. Recent theory has explored how animals integrate information from different sources and the conditions that favor the reliance on some sources over others. And this theory supposes that natural selection favors the evolution of integrator mechanisms that allow different sources of information to be similarly read by the genome. So we were interested in trying to study the integration of information from fathers and personal experience. And this is work that was done by Laura Stein when she was a PhD student in the lab, and she's now a faculty member at the University of Oklahoma. So Laura was interested in to see how stickleback fathers respond to predation risk. So remember, fathers are constantly having to defend their nest from predators and other forms of threat. And sculpin are a really common fish predator that love to specialize on raiding male stickleback nests. So she found that exposing fathers to predation risk causes them to change their parenting behavior. So this figure here is showing time fanning, that's on the y-axis here, over the course of the nesting cycle. So if you just look at the open bars first to start, as you can see, levels of fanning go up, 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 until right around when the eggs are about to hatch, and then they go back down again. When we exposed males to a model predator of this sculpin, Laura found that males decreased fanning in response to this simulated predation event. And this decrease in fanning persisted for a few days afterwards. And that's what's in the gray bars. So dads here are changing their behavior in response to predation risk. So when Laura reared up the offspring of these dads, comparing offspring of predator-exposed versus unexposed dads, she found that offspring of predator-exposed dads were smaller, they were in worse condition, they were less colorful and less active. And you might think from just looking at this list that predation risk to dads is bad for their kids. But interestingly, actually, these phenotypes that result from paternal predator exposure are similar to those that result from either evolved or developmental responses to predation risk, and being smaller and less colorful and less active might actually be adaptive if you're living in an environment with a lot of predators around. So which suggests the intriguing possibility that dads might be adaptively programming their offspring for living in an environment with predators. So feel free to ask me questions at the end about the mechanism by which this happens. Short answer is we don't know, but for the purposes of this talk, I can tell you that these effects are, we know they're not mediated by sperm, because in this experiment, fathers were exposed after fertilization. And we think it's mediated behaviorally. There could be olfactory cues that embryos are picking up on that their fathers are releasing, maybe through this crazy protein that fathers make to hold their nest together, possibly steroids that are excreted into the water. But we can definitely rule out direct offspring exposure because fathers were exposed before embryos had developed eye cups, so there was no way of the embryos themselves to see the predator, and fathers were exposed to a visual cue of a predator only. Okay, so now let's think about what happens then in the brain and in the behavior of a stickleback. If it's getting cues about risk from its father, what happens when additional cues or not about risk are personally experienced? So to get at this question, Laura did an experiment where she manipulated both personal and paternal experience with risk here to a sculpin predator, the same one she used in the study I just told you about. So she generated offspring from 10 fathers, 5 of whom were and 5 of whom were not exposed to risk, where they were exposed to risk 3 days post-fertilization, as in the previous study. And then she split those offspring within a family across 2 different treatments. Here, the offspring either did or did not get experience with risk. And in this case, they were chased by the model sculpin every day for a week, and this was meant to simulate predation pressure. So in this experimental design, Laura basically generated 4 different types of animals. Those who weren't exposed to risk themselves and their fathers weren't exposed, we're calling that sort of the double control. Those with personal experience of risk, but whose fathers did not have experience. Those with no personal experience, but whose fathers did have experience with risk. And then what we call the double whammy group, where offspring and their fathers were both exposed to risk. And then when the offspring were 3 months old, Laura measured a bunch of traits on them, including things like size, condition, behavior, sex, and brain gene expression. And she also used a male-specific marker to genotype them for sex. So here are just some of the results focusing on the comparison of offspring of unexposed versus predator-exposed fathers. So as you can see, offspring of exposed fathers were smaller and they were also more timid. More timidity here was measured by how long it took the fish to emerge from a refuge, so relative to offspring of unexposed fathers. And this is exactly the same pattern as what she found before in a previous study. So we're seeing consistent effects of paternal exposure. So just focusing now on the offspring of unexposed fathers, we can ask about how offspring that were versus were not exposed to risk themselves. You can see that offspring that experienced risk themselves were also smaller, and they were also more timid. So this pattern has been documented in other studies on sticklebacks and other small fishes, and it's been interpreted as an adaptive life history response to predation pressure. So if there's a lot of predators around, you might be better off, reproducing at a smaller size and getting it all over with because it's more likely, survival is less likely. So regardless of the source of information, whether it came from dad or from personal experience, sticklebacks are producing the same set of phenotypes. In other words, developmental and transgenerational plasticity seem to be two different ways of achieving the same phenotypic outcome. So everything here is behaving exactly the way we were expecting in terms of how personal and paternal experience with risk independently influence these phenotypes. But what about when these two are combined? So there was actually a significant interaction between offspring and father treatment such that the effects of both were non-additive. So the pattern I'd like to draw your attention to is that on the left, when dads weren't exposed to risk, there was an effect of offspring treatment. The means here are different. But here on the right, the effect of personal experience goes away. So offspring that got the double whammy, the ones on the far right here, are getting cues from both their father and from their own experience that the environment is dangerous. But it's not like they end up twice as small or twice as timid. They're the same size as predator-exposed offspring and offspring of predator-exposed fathers. So this is actually a little bit different from what some of the models predict, that we should see the most extreme phenotypes when information from different sources is in agreement. But here, if your dad was exposed to risk, additional personal experience didn't really add anything to the phenotype. This kind of result is actually, I think, more consistent with some more Bayesian type models that predict that when information is consistent with prior expectations or it's redundant, you should ignore it. It's only when you get new information about risk that an animal should respond. So offspring ignore personal experience when it's consistent with their prior expectation that was set by their father. Another possibility, of course, is that there could be something like a floor or a ceiling effect or other type of constraint on the phenotype that can be generated. But another way I think to think about this has to do with the costs of failing to respond to cues about danger. So stickleback are prey species highly vulnerable to predation risk. Therefore, they might be better off responding 100% to a false alarm than not responding at all. So the so-called smoke detector principle. So and this might especially be the case if the costs of mounting a response are relatively low. So the joke that I like to tell about this is that, so unlike Nemo, stickleback actually do listen to their dad, and they don't need personal experience to confirm what their dad has already told them, and they're good kids. So that's pretty cool, but recall that Laura also measured gene expression in the brains of these offspring. So what we're doing here is we're comparing the number of differentially expressed genes in the brains of offspring whose fathers either were or were not exposed to predation risk. In other words, this cell versus the double control. And there were about 900 or so DEGs in that comparison. The circle on the right is showing the number of differentially expressed genes in response to personal experience with risk. In other words, this cell versus the double control, which is also about 900. And the area in the overlap is referring to the number of genes that were in common between these two comparisons, which is 344. In other words, 344 genes are shared between these different forms of plasticity, and this is a highly significant overlap. It gets more interesting, though, when we actually look at the expression patterns of those shared 344 genes highlighted in pink. So this heat map is showing the expression pattern of the genes that were in common, where red refers to upregulated, blue downregulated genes when comparing offspring of predator-exposed versus unexposed fathers is on the left, and developmental plasticity genes here are on the right. And when Laura showed me this, I was like, oh, you must have made some error in your code because this pattern is way too congruent, never looks like this. Genes that are going up in response to father's experience with risk are also going up in response to personal experience with risk, and vice versa. So it looks like the brain gene expression profile of an animal that personally experienced risk looks very similar to the brain gene expression profile of an animal whose father experienced risk, or remarkable overlap at the molecular level between these two different forms of plasticity. So one way of interpreting these findings is that both forms of plasticity are sort of co-opting or activating the same molecular mechanisms, or that the genome is read in the same way by different sources of information. In other words, these different experiences, dad's experience or your own experience, are equivalent or interchangeable at the molecular level. And this concept of equivalence was discussed at length in Mary Jane West Eberhard's famous tome on phenotypic plasticity, but in the context of the equivalence or interchangeability of genetic and environmental effects. But here we're talking about equivalence in the context of two different environmental effects that are acting over different timescales within versus across generations. So what about, though, that other really interesting treatment, the ones that got the double whammy? So information from both personal experience and from their father that the environment is dangerous. So here what we're doing is comparing these individuals in the double whammy group to the double control. And the Venn diagram here is showing the overlap between all these different contrasts. I just want to draw your attention to the number in pink, which are the number of genes that were differentially expressed in response to personal experience, dad's experience, and the combined double whammy treatment. And the expression pattern of those 208 core genes is shown here with a new column here on the right, the both treatment. Notice that the brain gene expression profile of animals that got the double whammy looks the same as those that got just one source of information on its own. So to me, this result is suggesting that there might be something like a molecular switch that gets activated in response to information about risk, regardless of where that information comes from. And once that switch is on or turned on by dad, it stays on. And the pattern is also consistent, I think, with the phenotypic data that showed that the combined influence of personal and parental information about risk was concordant with either source of information on its own. So therefore, these brain gene expression data are consistent, I think, with the other traits. Personal and paternal experience are redundant. Okay, so what have we learned from this? We showed you that personally and transgenerationally acquired cues about predation risk triggered the same set of predator-adapted phenotypes. The combined influence of personal and paternal cues was non-additive, and personal and paternal cues were interchangeable at the molecular level, and we think we might have evidence for something like a switch. So with that, I realize I've gone on a few minutes too long. I apologize, but I would be more than happy to take any questions, and thank you very much for your attention.
Thanks, Alison, for the great talk. Really, lots of results and lots of things to think about. So we already have a couple of questions in the in the Q&R tab. So the first one is from Nicolas Proust. So Nicolas, you should be able to turn on your microphone now and state your question. Can you hear us? Is Nicolas still around? Well, the question, otherwise I can read it. I can read it, and Nicolas, please react if you want more detail. So he was asking, can we identify good and bad fathers by comparing gene expression levels? And do the expression of genes, are they associated with efficiency of behavioral, of the behavior?
Yeah.
So I guess there's something about looking more into quantitative traits, I would guess, something like this.
Yeah. So, I mean, that's sort of what we're trying to do now with this unusual ecotype that doesn't provide care. I mean, I think we could talk a little bit about whether this white ecotype that removes the eggs from their nest, are they bad fathers, or they just kind of have a different strategy. But we don't, so we're doing some experiments where we're crossing these two different ecotypes and using QTL mapping to try to identify parts of the genome that harbor genes influencing these different traits, so this dispersal behavior versus parental care. Don't have data on that yet. In terms of like just taking, say, a transcriptomic approach, you might imagine like if we could somehow compare fathers that provide a lot of care versus fathers that don't provide very much care. And it's true in all the data that I showed you today, none of this showed any individual variation. You know, that wasn't really the focus. But that's actually something that we're really interested in the lab, even though I didn't talk about it today, was that individual fathers differ dramatically and differ consistently in how they behave as fathers. So we've, even within the same population, we've got some dads that consistently say fan their eggs for longer compared to other dads. And this is something that is consistent about them. So if they breed another time, if they mate with a different female, that these seem to be relatively consistent attributes of them. When we actually did some quantitative genetic studies, just looking at parent offspring, using parent offspring regression to try to estimate the heritability of fanning behavior, we actually found that I'm like proud of this result and also a little bit embarrassed about this result, but the heritability estimate was 0.96. Which the reason why I'm like, I get embarrassed about it is because like, no, behavior is 0.96. That's ridiculous. That's like, that's impossible to be able to get that sort of estimate. But, you know, the data are what the data are. It's based on parent offspring regression, where we compared the fanning behavior of sons versus the fanning behavior of their fathers. There is a possibility of environmental paternal effects in that study because the sons actually received care from their fathers. So there could be some potential for learning there if sons resemble their fathers because they learn how to parent from their fathers, and the design isn't good enough to be able to tease that out. I will say that in other studies where we've tried to get at that question, we haven't found a lot of evidence for learning about caregiving. So, but anyway, that's my caveat about that result. But you were also asking not just whether there are good and bad fathers, but whether that, I think the question had something to do with if that maybe the expression could be correlated with their behavior. Oh yeah, can we identify good or bad fathers by comparing gene expression levels? Yeah, so we haven't actually done that at the gene expression level. We've got some quantitative genetic estimates on heritability, and we've got some ongoing QTL mapping things, but haven't actually explicitly compared gene expression between high and low parenting dads.
So Nicola doesn't have a microphone, so if you have some further questions, you can type them, I guess. And now we're going to move on to the next question by Frédéric. So just a second. Frédéric, you can turn on your microphone if you want.
Hello, can you hear me?
Yes.
Yes.
Hey, very great talk. Thanks very much. I've been looking at these genes that you mentioned in your comparison with the mammals, and among orthologs, there's the SOC3 genes that is not duplicated in fish compared to mammals. But what about the other genes that you found? Are they, because these genes have been through the Taylor's wall genome duplication, and some of them remain duplicated, like SOX genes, but not SOC3. But, tyrosine receptor kinase or nuclear receptors. How about the other ones that you mentioned?
Yeah, that's great. I don't know the answer to that offhand, but I can say I know that in the analysis, we used both one-to-one and one-to-many ortholog mapping to try to account for possibly duplicated genes, but I don't know if we've actually looked closely to see the distribution of the multiple orthologs and whether or not they were more or less, say, abundant in the shared genes. I'm sorry, I don't know the answer to that.
Okay.
Yeah.
Okay.
But that, yeah. Do you have a hypothesis?
Not yet, but if you want, I can have a closer look to these genes, maybe.
That would be sure.
We can talk about that later.
Yeah.
Okay.
Thank you.
So while we wait for further questions, I would have lots of questions actually, but well, people can type more questions in the Q&R tab. So what struck me about this latest point we discussed about this oxytocin pathway is that, if I understand well, this seems to have more effects in mammals than it has in fish, because it also affects physiology, etc. Right? So it would be the hypothesis would be that this pathway that was first involved mostly in behavioral traits related to parental care would then be co-opted and combine this with perhaps physiological aspects in mammals like milk production and things like that. Is that what you think? So the trait would have diversified. I'm not very familiar with what oxytocin does, but
yeah, it's crazy. I don't know. I don't have it.
It would have become some kind of maternal syndrome based on a more simplified or mostly behavioral trait. I mean,
yeah. So one of my favorite examples is prolactin, and prolactin is really interesting from a fish point of view because it's really important for osmoregulation. So many stickleback are actually anadromous, and they go back and forth between seawater and freshwater, and prolactin is a really key hormone regulating their ability to osmoregulate and change their ability to excrete water or hold on to salts as they're making that migration. And so there's actually like prolactin receptors on the kidneys, and levels of prolactin go up during migration, which is also the time of the onset of breeding, and levels of prolactin or higher levels of prolactin also seem to promote parental care. So like, but what prolactin is doing, so it's acting somehow on the kidneys, it's acting somehow in the brain, and they're seasonally correlated with one another. They're different prolactin receptors. This is haven't studied this at all, but it's one thing that it's one of the things that I think would be really interesting to try to understand more about how these really deeply conserved hormones can have these different functions. And yeah, you also mentioned about oxytocin. And
perhaps I got this wrong. I don't know if oxytocin also has physiological effects in mammals. I'm not sure. Perhaps I misunderstood what you say. I didn't remember well.
I mean, so oxytocin, I think, is sort of one of the canonical hormones that's sort of associated with lactation.
Yeah, that's what I thought also. Okay.
Yeah, yeah. And that's super interesting because oxytocin also is high while male sticklebacks are incubating eggs. And so, you know, the extent to which a father incubating his eggs is analogous to something like pregnancy, you know, in mammals. Yeah.
Right. Emilia actually has a question, I think. Emilia?
Yes, I have one question. Do the father and personal juvenile experiences of predation risk influence in terms of offspring care behavior?
Yeah. So do offspring of predator exposed fathers behave differently as fathers? Yeah. I wish I knew the answer to that. We have attempted that study, but have not been hugely successful in actually getting data to address that. I mean, the short answer is I don't know. That a sort of dirty little secret about stickleback biology and stickleback lab work is that generation time is a year. So to do these sorts of experiments, you know, these are multi-year types of experiments. Another thing is that we tend to study animals from natural populations, and so we can bring fish into the lab and they tend to behave really well. These wild-caught animals tend to behave really well in the lab, but the longer we keep them in the lab for multiple generations, they start to act funny. And it's harder to elicit reliable behavior from them. So anytime we have to get into, like, you know, the F2s or the F3s, things often start to deteriorate and become a little bit harder to interpret, not to mention it's like year 4 of the experiment. So I don't know. Do you have a hypothesis?
Yeah, it would be adaptive that the effect persist on the second generations. I think so.
Mm-hmm. Yeah, I will say that I didn't talk about this work at all, just focusing on the behaviorally mediated effects, but we have done some sperm mediated effects. So Jen Hellman was a PhD, or a postdoc in the lab, and she was able to keep them for 2 to 3 generations and to see whether or not any of these and any sperm mediated paternal effects persisted into the F3. And she does have some evidence for that. In that case, though, it's not a behaviorally mediated. It's not that the father is changing the way that he interacts with his offspring, but it's something through sperm.
Okay.
Yeah.
Right. So yeah, Vincent is the co-host, co-head of this network. Sorry, you haven't been introduced. So Vincent, you had a question?
No problem. I'm sorry, I was late for the talk. Sorry about that. It was really a great talk, really clearly explained. So I almost, I mean, at least I felt I understood most of it. So pretty happy with that. I had a basic question on the story in terms of adaptive evidence of this plasticity. Did you test whether the induced phenotypes in the offspring were actually adaptive relative to maybe predations? And so a related question, whether there were any costs of this plasticity, maybe I mean, in the absence of predators in the offspring, and whether there could be costs in terms of I don't know, sexual selection or something like that.
Yes, so those are the million-dollar questions, aren't they? Those are the things that we really would like to know. So in terms of adaptive significance, the way that we could kind of get at that in the lab is to try to simulate kind of real-time survival under the face of predation risk in the lab. So if we can have, really big tanks and we have offspring of predator exposed versus unexposed fathers, and we actually put, a live predator in them and then in the tank, and then we see who gets eaten. Those experiments have proven to be pretty hard to do for a number of different reasons, and they tend to be kind of artificial, just because in nature there's so much more complexity of the environment that can influence the outcome of a predator-prey interaction. And unfortunately, we don't really have the facilities, I don't think, to be able to do it well. I think to do it well, we want to put them in ponds, artificial streams or artificial lakes or something like that, that would allow prey to be able to use multiple types of predator defenses during their interactions with predators. I'm trying to think, I don't know, I'm trying to remember if we actually have any published data on some of these, because we've been trying to actually get some data on this, but I don't actually think we have any clear evidence that offspring of predator exposed fathers actually survive better. I mean, our inference has been that many of the phenotypes that are induced are consistent with evolved phenotypes in stickleback that happen to be subject to strong predation pressure, smaller size at maturity, less coloration, less colorful, faster early life growth, we think is adaptive for being able to escape size selective predation later on in life. I'm sorry, I'm not giving a great answer because I don't have a great answer to this question, other than to agree that I would really like to know the answer to that question. In terms of fitness costs, also, right, you mentioned about coloration. So it does seem like offspring of predator exposed fathers tend to be less colorful, and male coloration is sexually selected. It's important both for male-male competition as well as for female preference in this system. And so that could be an example of a cost in a sexually selected trait.
That's right.
Yeah.
I had actually a very technical question, something that I misunderstood, so others might have misunderstood. It may be about ChIP-seq that I don't really know, because in this first part of the talk, you were mentioning something about transcription factors. Is it that ChIP-seq allows finding places that are accessible to specific transcription factors? Is that how this works? Or because I could see that some promoters were more accessible, but I didn't see the transcription factor part of the story.
Yeah, so sorry, the transcription factor piece came in through the gene regulatory analysis. So I didn't talk about like the method for that at all, which is probably why it wasn't clear. But basically, our colleague collaborator, Sriram Chandrasekaran, has a really cool program that uses existing information about conserved transcription factors and their targets to build gene regulatory networks based on patterns of co-expression in the gene expression data. So we can use the gene expression data to find genes whose expression patterns are correlated with one another. We can also do things like then look upstream of those genes that are similarly correlated to see if they have conserved motifs for transcription factor that are, And
so
then
this is how you infer retrospectively that this transcription factor might have been involved because they have the same motifs, right?
Yes, exactly. But we were also able to then go into the data set itself and to look at the expression of those transcription factors themselves. And that's where it became pretty cool, because the expression pattern of the predicted targets of the transcription factors was actually correlated with the expression patterns of the transcription factors. And that was sort of reassuring that, like, okay, yes, this bioinformatic, who knows what's happening, is actually telling us what we could have predicted. And then we cherry picked a few cases to try to see, like, can we see whether there's parts of the genome that are becoming more accessible? So transcription factor binding sites that are differentially accessible based on the ChIP-seq data. And then, what are the transcription factors that should be binding in those differentially accessible areas? What are their predicted targets from the GRN? And then what is the expression profile of those predicted targets in the actual gene expression data? And we found a few examples where those things actually did align, which also gave us some reassurance.
Okay, thanks a lot. That clarifies it. Cedric had a question. Cedric?
Yes. Hello?
Yes, we can hear you.
Okay, thanks. Thank you, Alison, for the great talk. You seem to be willing to talk about these white males, and I like to hear about them. So are there two clear different tactics, or is there intermediate phenotype? How heritable is it? Do you see differences in gene expression or things like that?
Yeah, cool. The white stickleback, this is like the thing that I'm obsessed with right now, because they've
you have another talk ready with other slides at the end, right?
It is. I've got a whole other talk about the white stickleback. They're very, very cool. I mean, so we got interested in them because of all of these interesting things about parental care that we were learning. So we compare different populations and see different patterns of care. We can estimate heritability of care. We find, oh my gosh, it's really, really highly heritable. That suggests that that it's a trait that's potentially amenable to kind of more molecular and genetic dissection. And so we knew about these white stickleback had been described back in the 80s with these classic studies by Max Blau and other behavioral ecologists in Canada, who had observed that there is an ecotype. So these are not alternative strategies within the same population. These are reproductively isolated ecotypes. Sometimes they co-occur, so sometimes sympatrically, sometimes allopatrically. They seem to be restricted to Nova Scotia. And they see their divergence seems very, very recent. So estimates of genome-wide divergence are actually very, very low. But they look really different and they behave really different. So even at the genomic level, they do not seem very diverged at all. Phenotypically, they do really, really different things. So in allopatric populations, just for simplicity, it's useful, I think, to think about those. So we refer to the regular stickleback that occurs in Nova Scotia. We call those commons because they're kind of like the ancestral form. And the white ecotype is derived from this common form. We think very, very recently. So the common form does the regular good old stickleback thing, males are territorial, nesting territories, attract females to mate, do the whole long song and dance, care for embryos, care for fry, do that whole thing. And they have a pretty short breeding season, probably about, a month to 6 weeks or so to get it all done. The white ecotype, on the other hand, they build nests, they defend territories, they court females, they do the whole external fertilization thing. But then after fertilization, they pick up the eggs from the nest and they spit them out of the algae into the algae. These embryos seem to do just fine without their fathers, which, you know, here we've been studying like how important fathers are, and like kids need to learn from their fathers, and they get all this information from their fathers, and you know, they need to get protection from their fathers. But these white embryos seem to do just fine without their fathers, without being fanned or anything. And an original hypothesis for why maybe this white ecotype had evolved in this area is that there's a lot of filamentous algae in some of these estuaries where the white ecotype is breeding. And so one hypothesis was that, oh, well, you know, the embryos, maybe they're getting protection by being in this algae, and maybe they're also getting aeration from the algae. And so that liberates the father from having to care for them because the embryos are getting aerated and defended by the algae. I don't think that's necessarily true. We don't have really, so in lab studies, we can rear them just fine without the algae, and we see the white ecotype in areas without the filamentous algae. So I don't think that's necessarily true. I don't think it's the driving, you know, the driving force behind the evolution of this interesting divergence.
So the nest here is used only for sexual selection purpose?
Yep.
Is it very different in shape than the nest where of the common form?
Yes.
Yeah.
It is. Let me see if I can. How do I get out of here? Oh, here we go. I can. I might not be able to. I might be able to pull up some pictures. If you don't mind, I can show you something else. This is cool. Here we go. Screen share. Let's see. Can you see this slide? Okay, so this is a male common. This is a male white. This is a picture of a male common nest. So this is the typical stickleback thing. They collect algae, they kind of pound it down into a nice flat mass, and then they make holes in it, and then they burrow a tunnel through the middle of the nest. And then that's called this creep through behavior. Female swims through the tunnel, he fertilizes the eggs, and the eggs grow up in here. So this is what a typical common nest looks like. It's pretty flat. It has a lot of algae on top of it. And it also has a hole that goes through the side. This is what a white nest looks like, which is like this pile of algae that he sort of makes this hole on the side through, but he doesn't really use any sand, and he doesn't flatten it down at all. So we can, you know, quantify these things like how tall is it, how much algae or how much sand is used on it, whether the opening on the nest is on the top or on the side. And so we came up with this nest score just to try to quantify these different dimensions. And this is what the nest score of the common ecotype versus the white ecotype, and we've got some F1 hybrids and also some F2s. So this, I think, actually, this nest architecture could be really interesting, I think, from a genetic point of view, because it looks like there's some nice variation that can be segregated in the F2s.
Oh, great.
I mean, I love the nest stuff too, because nests are probably sexually selected in this group. So females pay a lot of attention to nest architecture when they're making mate choice decisions. Also, when you look at the courtship behavior of commons versus whites, commons show off their nest a lot during courtship. They lead females to the nest and fan at their nest during courtship, even if they're not providing care. And it's thought that this is supposed to be like a reliable signal of parental ability. He's like, look at me, I'm going to be a great dad. Look, I'm so great at fanning. And then one of the things I really love about the system is that the male whites will also fan their nest during courtship, but they never actually go on to fan the developing.
So the females, they also must be plastic in their response to the different males, or well, maybe we will go too far on this, but it's ready.
Yes, also I'd like to say that Alison said she could perhaps stay for another perhaps 30 minutes if people want to have a private chat with her, right?
Yes.
Still, I want to take the opportunity that you're still here with everyone, because I wanted to just kind of a question that sits at the core of your talk, and I think it's really interesting. It's rare that you have an opportunity to see this addressed so directly. Is all these time scales of plasticity. You know, in this first thing, you saw that 5 minutes exposure leads to a lasting effect and things, and then, but at the end, you said. But the reality is that in the field they have all this, you know, contest or I don't know, the negotiations, etc. And in the other case, it was, you know, that yeah, I mean, some things that were carried out across generations and things. And each time I see something like this, I think, wow, this is fascinating, and this, but sometimes also makes me pessimistic because it's so difficult to integrate all these timescales. So, do you think there are some we can try to find some general patterns? Like it seems like you've seen some things that are a bit like a trigger, for instance, regardless of whether it's offspring or parental thing, you get the same trait, and then this sort of reduces dimensionality in a way. There's just one response could be triggered by perhaps some traits are not like that. I don't know, what are your thoughts? I know it's a bit difficult or vague, not very precise question, but you see where I'm trying to.
I do.
Yeah.
I do. I know, and I do not have an intelligent response to that question, other than it's something that I think about. I also think about a lot too, because I mean, so one thing I've been thinking about a lot lately is that when we do these sorts of RNA-seq studies, we the work is always in the data analysis part. Like it's very easy actually to generate, I mean, not very easy, I don't want to say it's like trivial, you know, to do these experiments and bring them into the lab and do careful behavior stuff. Like that's, I don't want to minimize that, but the majority of the work is spent in actually the bioinformatic downstream analyses of all of the data, and we just have too much of it. And so, and it's also very, very expensive to generate. And so when we're trying to figure out like what should be when we're interested in traits that are highly dynamic and where the expression of genes that are important for the trait strongly depends on the tissue. And when we're talking about the brain, I mean, this isn't liver, you know, this isn't like a muscle. It's not like a liver cell is a liver cell, nothing against livers or anything, but brains are very, very complicated. I am not a neuroscientist, but I know enough to know that what you measure strongly depends on where you look. And so having to make choices about spatial specificity, temporal specificity, how do you figure out what matters, and how do you do that without like spending gobs and gobs and gobs and gobs of money doing everything and getting way, way too much data to be able to figure out like, oh, this is the right time point and this is the right brain area that's going to tell me what I'm really interested in. Yeah,
it's difficult, but my question was also related to what you were talking about, integration of cues, because it seems like in I can't remember which examples it was, like the programming of predation risk. It seems like it's like a trigger or threshold or something, and then well, regardless of who is exposed at what time, you get the response. Perhaps in the first example, you were suggesting also something like that just a single encounter would trigger this learning. And then, I don't know, then this sort of simplifies the problem in a way, if we can make sense of it in this way. So,
yeah, I mean, another really fundamental constraint with when we're talking about profiling in brain tissue is that it's terminal. So it would be great. So we can't get repeated samples on the same individual. So it would be great, you know, if we could measure what's happening in an individual's brain at 30 minutes, and then again, look at what's happening at 60 minutes to actually look at the effects of experience, you know, within an individual. And because it's terminal sampling, you know, we can't get at that.
Okay, but well, anyway, this was a really, really stimulating talk. I think lots of people would probably, I wrote down lots of things about it. So I want to thank you again for this. And so I encourage people who still have questions to write to you, probably, as you said, and perhaps you have a little more time now to chat.
Yeah, and I would be happy also to meet with people. I know this is the end of the day for you there, and so, you know, it's like past 5. So I'd be happy to meet with people another time too.
All right, so thanks a lot. And with this, I'd like to say goodbye to everyone. see you for our next webinar. Bye-bye.
Thank you.
Bye-bye.
Hi, thank you very much, Alison. It was very great.
Thank you. Yeah, thanks for having me.
Yeah.